Coming to our next chapter, plasma proteins. So in plasma we have proteins, and coming to the concentration. So before going to details of the concentration of total proteins in plasma, so what is plasma? Plasma is blood minus cells, so that is called as plasma. And coming to serum, what is serum? Blood minus cells plus coagulation factors is called as serum. So in plasma you have proteins and their concentration together, we call it a total plasma concentration, and its normal range is 6 to 8 grams per deciliter. Right? So in this we have different proteins. The major plasma protein is albumin, and it accounts for 3.5 to 5 grams per deciliter. And coming to albumin, its concentration is 2.5 to 3 grams per deciliter, 2.5 to 3.5 or 3 grams per deciliter. And coming to the fibrinogen, and its concentration is 0.2 to 0.4 grams per deciliter. So among these three proteins, these three are called as plasma proteins, and their concentration is 6 to 8 grams. Major plasma protein is albumin. Yeah. And the Ag ratio, normal albumin to globulin ratio, is 2:1. And these plasma proteins can be separated by different methods, and the three common methods are salting out, Cohn's fractionation, and electrophoresis. So these three methods are used for the separation of plasma proteins. So what is salting out? So we use different salts to separate the proteins, like ammonium sulfate, right? So that ammonium sulfate is used for the separation by a process called as precipitation. So salting out generally uses precipitation so that proteins can be separated. So albumin can be precipitated by ammonium sulfate by full saturation. When you take globulins, they can be precipitated by half saturation. So different salt concentrations are used for the separation of plasma proteins. So this method where salt is used for the separation of proteins is called as salting out. And coming to the second method, Cohn's fractionation. In Cohn's fractionation, different concentrations of ethanol are used at various temperatures. So ethanol at various temperatures is used for the separation of plasma proteins, and that method is called as Cohn's fractionations. So different fractions of proteins are obtained in this method. So fraction 1 to fraction 5. So we'll get different fractions. And coming to the significance of Cohn's fractionation, so this Cohn's fractionation is used for the separation of proteins at large scales. So the proteins used for the therapeutic uses, they are separated by using this method called as Cohn's fractionation. Coming to the third method, electrophoresis. So here the proteins can be separated using this method called as electrophoresis, where the separation is based on the movement of charged particles under the influence of current or electric field. So this method of separation of proteins based on their charge in the presence of current or electric field is called as electrophoresis. Okay. So by electrophoresis, proteins can be separated into different bands. Those are albumin, alpha 1, alpha 2, beta and gamma globulins. So we'll get different bands on the electrophoresis, and the fastest moving fraction in case of electrophoresis is albumin, and the gamma globulins lies at the site of application of sample. The slowest moving fraction is gamma globulin, and the fastest moving fraction is albumin. So we'll get different fractions, 5 fractions when you subject proteins to electrophoresis, plasma to electrophoresis. So that is albumin, alpha 1, alpha 2, beta and gamma globulins. Coming to functions of plasma proteins. So what about the proteins in the plasma? They have different functions. For example, they are transport proteins. So like albumin. So albumin is a transport protein. It transports different hydrophobic compounds present in the plasma blood, like it transports the drugs, it transports bilirubin, it transports other hydrophobic compounds like calcium. So this albumin acts as a transport protein because it is involved in the transport of various hydrophobic compounds, so to the target tissues. Likewise, we have thyroxine. It is transported by a protein called as transthyretin. And prealbumin, it also transports thyroxine and triiodothyronine, right? And transferrin, a iron transport protein. So like this, we have different proteins, plasma proteins, whose have a role in the transport of different molecules. So that's why we call them as transport proteins, and that is one of the functions of plasma proteins, transport function. Coming to nutritive function. So when you take albumin, it is a complete protein because it has all the essential amino acids and the required proportions. And this albumin acts as a source for the essential amino acids. So those essential amino acids cannot be synthesized in our body, but they are required for the synthesis of various proteins by the target tissues. So in those target tissues, wherever they require these essential amino acids, and the source for these essential amino acids is albumin. So albumin transports all the essential amino acids to the target tissues. So in this way, plasma proteins, they have nutritive function, and the example is albumin with all the essential amino acids. So we call this albumin as a complete protein. Coming to another function, buffering action, and this we discussed in case of acid-base balance that the first line of defense is brought by the buffers. We have different types of buffers: bicarbonate buffer, phosphate buffer, and protein buffer. So protein buffers. So here plasma proteins, they have the buffering action, and the major plasma protein is albumin. So most of the buffering action or capacity of the plasma proteins is because of the major plasma protein albumin. And proteins, they have buffering action due to presence of a particular amino acid called as histidine. Each albumin has 16 histidine residues in molecule in one albumin molecule. So when coming to the other protein which has the buffering action, hemoglobin, that also has histidine residues. Each hemoglobin has 38 histidine residues. So because of presence of histidine residues in these proteins, that is albumin and hemoglobin, they show buffering action. So they can regulate the acid-base balance, they can regulate the pH of the blood. Right? So that is the buffering action of proteins. Coming to the colloidal osmotic pressure, another important function of plasma proteins. So this is shown by the plasma proteins, and again coming to the major plasma protein, that is albumin, acts as a, so it creates a pressure called as effective osmotic pressure. So there are two types of pressures exerted in the vascular compartment, that is blood pressure and effective osmotic pressure. So this effective osmotic pressure is because of the plasma proteins. These plasma proteins, they cannot move out of the vascular compartments. Because of this, they create a pressure called as EOP. So in short, we call it as EOP, effective osmotic pressure. So according to Starling's hypothesis, at the arterial end of capillaries, so the effective osmotic pressure is 25 mmHg and the blood pressure is 35 mmHg. And here the difference is 10 mmHg, so that the water enters into the tissues with a pressure of 10 mmHg. And coming to at the venous end of capillaries, the effective osmotic pressure is 25 mmHg and the blood pressure is 10 mmHg. So water is imbibed into the tissues with the pressure of 10 mmHg. So water comes into the vascular compartments with a pressure of 10 mmHg at the venous end, and it is expelled at the arterial end with a pressure of 10 mmHg. So this maintains the balance between water expelled and water imbibed. So because of that, it maintains the fluid balance. So because of the presence of the major plasma proteins like albumin, it creates effective osmotic pressure, and that is responsible for this pressure called as colloidal osmotic pressure. So whenever there is decreased albumin concentration, it affects the effective osmotic pressure. So that's why in case of hypoalbuminemia, whenever there is decreased albumin concentration, it leads to edema, because the pressure with which the water is imbibed will decrease. So that leads to retention of fluid in the tissues. That condition we call it as edema. So edema is because of the hypoalbuminemia, so decreased albumin concentration. Coming to the other function, these plasma proteins, they act as antiproteases. So they act against the proteins. So there are some proteins like elastin, thrombin, plasmin, chymotrypsin, trypsin. So all these proteins, they have serine in their active center, that is active site. They have a functional residue is serine. So that serine is attacked by these proteins called as antiproteases. So these proteases, antiproteases, they act against the proteins. That's why we call them as antiproteases. So that is the other function of plasma proteins. Coming to another function, blood coagulation, we have different factors which are involved in the blood coagulation, another function of plasma proteins. Coming to hormones, so there are some hormones, protein hormones present in the blood like insulin, glucagon. They are nothing but protein hormones. That is the another function of proteins. They have hormonal function. Coming to immunity, so gamma globulin fraction of plasma proteins, they have the role in immunity. So that's why you call them as immunoproteins or protective proteins. Immunoglobulins are called as protective proteins. So plasma proteins with the fraction, that is gamma fraction, has role in immunity. That is another important function of plasma proteins. Coming to another function, so there are some proteins which are elevated in the inflammatory responses. So that's how the proteins are also involved in the another function called as inflammatory responses. Right? So these are the important functions of plasma proteins. Coming to acute phase proteins. So what are acute phase proteins? In case of some inflammation and neoplastic conditions, the levels of some substances, some proteins are elevated to by a concentration of 50 to 100, 50 to 1000 folds. So actually their concentration is very low, but because of inflammation conditions, the level of such compounds is elevated by 50 to 1000 times. So such type of proteins whose concentration is elevated in inflammation conditions is called as acute phase proteins. Right? So that's about the acute phase proteins, why you call them as acute phase proteins. Coming to the examples of acute phase proteins, we have C-reactive protein, alpha-1 antitrypsin, haptoglobin, ceruloplasmin. So all these are the acute phase proteins whose concentration is normal in case of their concentration is elevated in case of inflammations. Otherwise, they will be in the normal concentration in the blood. So that's about the acute phase proteins. And coming to immunoglobulins, so we have discussed that when you subject plasma proteins to electrophoresis, we will get different bands. One of the band is gamma globulin. So that gamma globulin fraction has the immunoglobulin function. So immunoglobulins are present in the gamma globulin fraction of the plasma proteins. Coming to immunoglobulins, they are involved in the immunity. They are the protective proteins. They protect the organisms from the invading microorganisms and pathogens. Right? So when you see the structure of immunoglobulins, it has 4 polypeptide chains, 2 light chains and 2 heavy chains. Right? So and it resembles a Y-shaped structure. And so when you see the structure, so this light chains and heavy chains of immunoglobulins, they have 2 parts, that is, one is variable region and the other one is constant region. And the variable region is towards the amino acid, constant region is towards the carboxyl end of the polypeptide chains. So each light chain of immunoglobulin has 220 amino acids, whereas the heavy chain has 440 amino acids. Right? And when these immunoglobulins are subjected to degradation by an enzyme called as pepine, so that pepine converts or degrades that immunoglobulin into 2 fractions, FAB and FC, right? Antibody fraction and crystallizable fraction. So those 2 fractions are obtained by the action of pepine on the immunoglobulin. And all these chains, light chain and heavy chains, are linked together by disulfide bonds. Interchain disulfide bonds are made. So they are responsible for the association of all these light chains and heavy chains in the immunoglobulin. Coming to types of immunoglobulins, there are 5 types of immunoglobulins: IgG, IgA, IgM, IgD and IgE. So based on the type of heavy chain present in them, we classify the immunoglobulins into 5 types. If the heavy chain is gamma, we call it as IgG. If the heavy chain is micro, we call it as IgM. If the heavy chain is delta, we call it as IgD. If the heavy chain is alpha, we call it as IgE. And if it is epsilon, we call it as IgE. So IgA is alpha, IgE is epsilon. So that's how, depending on the type of heavy chain, we classify the immunoglobulins into 5 types. And when you see, so that IgG, IgE and IgD are monomers. They have a single Y-shaped unit, so that's why we call them as monomer. And when you see IgA, it is a dimer. It consists of two Y subunits linked together by a protein called as a J chain. So that J chain binds these two monomers. And coming to IgM, it is a pentamer having the 5 Y-shaped units, 5 subunits. So all the 5 subunits are linked together by J chain. So this is about the classification of immunoglobulins into 5 types. And coming to the each class, first coming to IgG. So what is IgG? So G stands for gamma chain. Whenever the heavy chain is gamma, we call it as immunoglobulin G, IgG. And coming to its concentration, 75 to 80% of the immunoglobulins in the blood, they are of this type, IgG. Right? So it is the major immunoglobulin in the plasma, and it is a major antibody in the secondary immune response. Right? So that is a very important point regarding IgG. And there are different subclasses of IgG, and those are IgG1, G2, G3 and G4. So there are 4 subclasses of immunoglobulin G, and that depends upon the variation in the amino acid sequence in the heavy chain. So that is responsible for the subclasses of immunoglobulin G. And this is the only immunoglobulin that can cross the placenta and give the immunity to the fetus. Right? And this IgG is also involved in opsonization. So what is opsonization? Whenever a microbe comes into the body, enters into the body, this IgG binds with that microbe, and the Fc fraction of IgG binds with the macrophages. So this helps in the phagocytosis, and this type of process we call it as opsonization, and to be specific we call it as IgG opsonization. So that's about the functions of IgG. Coming to IgA, it is a secretory antibody. So in body secretions, we have this type of immunoglobulin, IgA. It has a heavy chain of alpha type, we call it as IgA. And coming to body secretions like saliva, tears, gastrointestinal secretions, right? And secretions of the lung, and all these urogenital secretions. So all these secretions, body secretions, they have this type of immunoglobulin called as IgA. So that's why we call it as a secretory antibody, right? And this has a monomer, or it may exist as monomer or dimer. If it is in dimer form, that is joined together by a J chain. Right? Coming to another immunoglobulin, IgM, having the micro type of heavy chain, so that's why I call it as IgM, and it is a pentamer. So it is a macroglobulin because of its heavy structure. Why it is heavy? Because it has 5 monomer units joined together by J chain, and this is the major antibody of primary immune response. So we have seen that IgG is the major antibody of secondary immune response. Likewise, we have some antibodies in the primary immune response, and that antibody is IgM. Coming to IgD, so it is present in very less concentration in the plasma, and there is no specific identified function of this IgD, but it is present on the surface of the B lymphocytes. Right? So this is about the IgD. Coming to the IgE immunoglobulin, it is a monomer. It has only a single Y-shaped unit. And coming to its function, so this IgE is involved in the mediation of allergic reactions, anaphylactic reactions. In all those reactions, so this immunoglobulin IgE is involved. So hypersensitive reactions. So this mediates all those reactions, allergic reactions, hypersensitivity, and the anaphylactic reactions. And this IgE binds with the subgranules monocytes and releases a compound called as histamine. So degranulation results in the release of histamine from the mast cells. So this histamine mediates the allergic reactions. So this is about the functions of IgE, which mediates the allergic reactions. So till now we discussed of the plasma proteins. What are the different fractions of plasma proteins, how they are separated, what are the functions of plasma proteins, and the important functions of immunoglobulins, so different immunoglobulins and their functions.